So I will switch to English now. Our guest of today is my pleasure to introduce him. He was very kind to come down from Florida to present his work to us in person and not online. So I'm very grateful. Thank you so much for joining us. Dr. Srinivasa Subramaniam is an associate professor in the Scripps Institute in Florida. He obtained his PhD in 2004 from the University of Heidelberg, Germany, where he worked on the molecular signaling that triggers neuronal death. So all his career, he has been very interested in studying neurodegeneration and cell death. Later, he went to Johns Hopkins to do his postdoctoral research, and he addressed the mechanisms for tissue-specific dysfunction, focusing on Huntington's and Parkinson's disease, investigations that he carries out now as an independent researcher. So his hobbies involve running, boxing, and sketching, and he actually had a couple of collaborations with Dr. Tapia, and they published a very nice PNAS paper. So today he will talk about these cool mechanisms to transmit proteins from neuron to neuron that are the nanotubules. So thank you so much for joining us. Welcome.
Hear me? Yeah. Oh, I'm very. I've been interested. Yeah, I want to go to Mexico, but then when Uri and Vayalta called me, I said yes, I'm going to go now. It's a great opportunity to see, also to see Mexico City. Always imagine that. So what I'm going to talk today is about how the protein can move from cell to cell physically and using a membranous tubes, and what that kind of transfer can have an impact on neurodegeneration. That's my major idea. But I would like you to be proactive. You can stop me if you have any questions during the talk and clarify certain things before we go on to move on to different slides. So I just wanted to add a point about Tapia. Actually, I had a great opportunity to collaborate with Dr. Tapia through Uri, is a faculty also here, and we've published a paper together. It's wonderful to have a collaboration with Tapia, excellent scientist. I'm sorry that he passed away. I was quite shocked. I thought I'll meet him one day physically, but I could not. But anyway, so now we all agree that we as a human, we communicate to the world through our brain. I think we all agree on the idea of communication. Why it's switching off and on. No? Nothing. I did nothing. Maybe something happened here. Computer died. Okay, open that. So I hope I can go after 2 slides, right? Okay. So we all agree that the brain works through these major senses that we have, right? Can't hear me? Oh, this is the problem. Can you hear me now? No? No, no, they said no. They can't hear. Can you hear me? The last. Hello. Now? Ah, okay. Yeah. Hello? A little bit up, up, up. Hello? 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 Okay. So we already see that these senses, the way our hearing is one of the senses, right? So it's so important. Our senses has to function to get the data from the environment and process that through the brain and interact with it. That's so fundamental, right? All this happens because we have neurons in the brain um, that makes these functions perform beautifully. That we don't realize how they perform so precisely through using these senses. I like this comic. This comic came out this year, and in that comic they say that everything begins and ends in the neuron. So I just leave it to you to decide on that. So, but this beautiful brain is affected by so many different diseases, which is listed here. You can imagine that these are all the diseases which affect everyday different humans in different forms. And we don't understand how this happens. We don't know effective therapies for any of these diseases. We don't have any effective therapies. This is a major problem. But in the future, we're going to live longer time, and then we are going to experience many different kind of diseases affects brain, um, and which is a major problem. When you start your senses not performing well, you're not able to interact with the world. So that's why it's important that we understand this process in the brain better, how the brain functions. So in my lab, what we do is we use different kind of tools, available tools, to understand the brain functions, our biology, and how it's impacted in diseases. So we use cell models, we use system approaches, we use mouse models. Sometimes we also use the monkey model, very rarely. So we take an integrated approach to address this complex brain problem. So what is that we do? One of the questions we address is that why in neurodegeneration only a specific part of the brain degenerate. For example, in Parkinson disease, the substantia nigra neuron degenerate. In ALS, the motor neurons degenerate. In Alzheimer's disease, the pyramidal neuron degenerate. I'm not doing anything. So my computer. Maybe it is something to do with. I can restart again. Don't worry. I think there is display issue. I think. There is some issue with the. What is the issue? Please, please don't go. I have a lot to. I've got a lot of things to say. I think you will like it. Yeah. What about you change that to this? Maybe there's a problem with that connection. Then what is the problem? Yeah, it worked, but okay. Yeah. So the major problem that we have currently in the field is that why certain part of the neuron degenerate in different neurodegenerative diseases. I mentioned about these three. For example, in FTLD, there is a neurons in this region called Van-Equon neuron they degenerate. In Huntington disease, which I'm going to talk more, well, the medium spiny neurons in the striatum degenerate. So we still don't understand why this specific set of neurons degenerate. That is the major question, is remain unresolved. And why to study this kind of study? As I mentioned, we are going to live longer, and there are many of this is going to come back, affect us, and we need to have a therapies to really resolve these diseases. If not now, at least in 20, 30 years, and effective therapies are important for a better human life. So, what I'm going to talk today is related to Huntington disease. I'm going to run this video in a second. So, what you see here is obviously we can identify who is a Huntington disease patient. So, here is an adult onset Huntington disease patient. You can see that some of the fundamental motor functions, like sitting quietly, or holding something, or driving a car, all this controlled by your brain function, particularly the striatum of the brain plays a very important role for this controlled movement. But when this controlled movement is disrupted, you get to this uncontrolled action that you will not have control about your own movement. We take it for granted, when you get up early in the morning, go brush, right? Brushing teeth. That's such a very complicated mechanism. But we don't pay attention to it. We just go on because it happens to us naturally, normally. But for Huntington disease patient, they can't do it. They need somebody to help them to brush. So we know that this is a genetic disorder. So we know the gene that is involved in this disease, which is called Huntington. So gene has been discovered quite some time ago, and as you can imagine, a lot of studies has been done on this protein, and this is a large protein in the brain, more than 66 exons, around 3,000 amino acids. And what happens is there's an N-terminal part of the protein just after 19 or 17 amino acids. We have got this CAG repeat, the gene that codes for glutamine. Normally, normal population of human, they'll have up to 35. But the adult onset Huntington disease patient, they have around 36 to 75. But the juvenile Huntington onset patient, they'll have more than 50, up to 120. So the difference is only this polyglutamine, the CAG repeat in the N-terminal part of the protein is sufficient to cause Huntington disease. And it is 100% penetrant. That means the people have this expansion in their gene, they 100% get the disease, no escape. Okay, what is the pathology of the disease? What happens in the brain of this patient? One thing is, this is an MRI of a healthy human. You can see this region is a striatum. This is the lateral ventricle. There's the striatum. There's a cortex, brainstem, and the cerebellum. If you compare that with Huntington disease patient, the striatum in this patient almost gone. They have a pathology in the cortex, shrinkage, but the cerebellum seems to be fine. Not much impacted. And the symptoms they get, the symptoms they have is well correlated with the striatal loss. Striatal shrinkage causes increased involuntary movement. You cannot control your movement. Huntington disease patient cannot drive. They cannot have control on driving. So that's the major pathology. So the question is, the mutant Huntington is present everywhere in the brain. present in the cerebellum, the brainstem, and the cortex striatum. But why does the striatum degenerate first? It is well documented that the striatum degenerates first, and then the cortical degeneration might happen. It's called a striatocortical degeneration, is observed in most patients. But the cerebellum seems to be okay, no problem, even though they have Huntington there. So that is the question. I like this one to emphasize the point. What you see here is the same patient who did been undergone MRI for 4 years, the longitudinal MRI. You can see the striatum within 4 years shrinks quite a significant amount of loss. Some little happens on the cortex, but the cerebellum seems to be fine. So this patient clearly show with the symptom associated with the motor symptom associated with the loss of striatum. Okay, when one striatum is gone, this is the striatal region. They have two major neurons in the striatum called D1 medium spiny neuron. D1 means they have a dopamine receptor expressed in medium spiny neuron, and there's the D2 medium spiny neuron. Both degenerate, and when that degenerates, all this connection from the cortex is gone, and the striatal to the other regions is also gone. So the striatum is critical for executing many functions. Suppose you decide you want to have a dinner tonight in a restaurant. You plan that, right? You plan all this in the cortex, and your decision should happen in the striatum. Okay, now I'm going to have dinner. All this planning, decision making, motivation, reinforcement all happens in the striatum. But when the striatum is not there, you can't perform this function. So most of the patients from the disease onset, all of them die within 10-15 years. Premature death. Again, the question, my point here is that why does Huntington affect striatum? That is my major point. Again, we're not the only one working on this problem. There are many people work on this problem, great scientists who spend their lifetime addressing this question. And there are many pathways have been described, like aggregation, Huntington aggregation, autophagy, G, mitochondrial dysfunction, transcription dysregulation, apoptosis, hepatotoxicity. All have been described in the literature. But again, why striatum? That's not very clear. So that is where the protein that we work with come into play. It's called RAS homolog enriched in the striatum. So this protein is highly enriched in the striatum, as you can see here. This is a mouse brain, very abundant. There is some in the cortex. What you're looking at is the mRNA expression. And look at the protein. This is expected molecular weight. This protein is highly modified in the striatum. This is the knockout control. And then if you look at the predicted structure of this protein has got a membrane binding domain, which uses the cysteine 263, and then it has got GTPase domain, a small GTPase domain. And what we found that this protein also has got like E3-like domain. And we have shown that this protein is modified at least 4 different sites by small ubiquitin-like modifier. This kind of shown here. Even in the human brain, this protein is highly expressed in the striatum. So the pattern is highly restricted to the striatum, a little bit in the cortex as well, in other places, but expression is highly abundant. Okay, so long story short, how did we end up connecting this protein to Huntington disease? There's 15 years of research. I don't have time to go through all that, but I'm going to give some kind of important slides with data before go and address Huntington disease. So the first data that came to us is some of the work that Uri was involved, Dr. Uri. So what we know in the field is that there is this plant protein. I'm sorry about that. There is this plant toxin called 3-nitropropionic acid, the mitochondrial complex inhibitor. when this is produced in the fungi, when this compound is injected in mice, what that does is it goes and causes lesions in the striatum. The striatum is degenerated for some reason, not the cortex. The striatum gets degenerated. And here is an example showing that this is a wild type mice, and this is a rest knockout mice where we injected 3NP intraperitoneally, and we looked at the structure. You can see that area, the lesion area is expanded here. This is the lesion area. Lot of pycnotic nuclei can be observed here. And this is a knockout. The rest knockout does not produce lesion as expected. If you plot the lesion area, you can see these are the wild type mice. Most produces lesion, but the knockouts, not many produce a lesion at all. Very little, if at all, very little. And this kind of shown here. And then we know we did more research on that. This way I collaborated with Tapia, Dr. Tapia as well. What we identified is that the rest identifies the damaged neuron, damaged mitochondria, and then interacts with the damaged mitochondria and then tries to remove them from the striatum. So 3NP damages the mitochondria in the striatum, and then rest goes and binds to the mitochondria and removes them from the striatum. What happens is there's the excessive mitophagy that causes lesion. Here is a photograph showing that there is the mitochondria surrounded by a rest protein. So we're working on this model, and we kind of surprised that now we know why the striatum degenerates in presence of 3NP, right? So this model also has been considered as a Huntington toxin model of Huntington disease, because the 3NP affects the striatum, which kind of mimics Huntington disease kind of phenotype. So it's been considered as a toxin model. So from this data, we went on to identify, okay, maybe there is some link with the genetic model of Huntington that we need to explore. Again, this work has been published, as you can see, a long time ago. I'll just go very, very quickly here. So what we have shown is that when you culture a cell, they do not normally have rest. So if you have a cell which have Huntington, they don't die. But then you put cells with the rest and Huntington together, the cells will die. That is kind of shown here. So clear loss of viability, and you can start seeing rest and Huntington cells, they form a lot of pycnotic nuclei. And this is kind of shown using FACS analysis, like the number of cells died in presence of rest in Huntington disease cells. So clearly there is a loss of cells when there is a rest and Huntington together, mutant Huntington. Then we did a PC12 cells. They do express endogenous rest. Okay, go ahead. Thank you for the clarification. Now, so what we have done is here, this is exponential cell. We just have a control vector, rest alone, control vector. Then we express wild type Huntington alone, nothing happens. Then mutant Huntington alone, not much happens. Then wild type Huntington plus rest. Then mutant Huntington plus rest here. So we do express in this wild type and mutant Huntington. And the same thing here, these cells, they have, as a wild type cells, they have Huntington with the 7 glutamine. And these striatal cells have a Huntington with 111 CAG repeats. So when you express rest here in the control cell, not much happens. This is a cell death indication, but the cells die when you put them in Huntington disease cells. So here in PC12 cells, when we deleted endogenous rest and mutant Huntington background, the cells start surviving better. So this kind of established that the basis that, okay, there is some connection between mutant Huntington and rest. We need to pursue further to identify the mechanism. Again, we spent a lot of time identifying the mechanism. One thing came out of this study is that normally the mutant Huntington, when expressed in the cell, they form aggregates, protein aggregates. Because of the expanded Huntington, expanded glutamine, they somehow gain this property of making aggregates in the cell. That's what is shown here. The aggregated Huntington and the soluble Huntington. Then when you express rest in those cells, what happens is that the reverse thing happens. They have more soluble Huntington and less aggregated Huntington. Okay, why this is important? This is important because in vivo, in the human, what they've shown is that the cells, they don't die, they have a lot of aggregate. But the cells, like in the striatum, the cells which die, they have a least aggregate. So there is some kind of biochemical property change happens in presence of rest. One of them is actually, obviously, there is a change in the soluble form of Huntington, which could be toxic. And that's why the striatum degenerates. We did a lot of work and then we showed the mechanism that the way it happens is that what rest does is it brings middle Huntington and another protein, which is called E2 ligase, sumo ligase, together and start enhancing the addition of another small protein, out of protein here, small ubiquitin modifier onto middle Huntington. When rest increases the simulation of middle Huntington becomes more soluble. And that's the property that rest is doing in the striatum. Then if you remove the simulation site on the Huntington, the cells won't die. And the aggregation property is reversed. So we got this strong biochemical evidence that how rest may be modifying Huntington and interfering in its biochemical property and then promoting toxicity. Well, they're all cell culture model, right? Then we want to do that experiment in knockout study, knockout mice. Go ahead. Yeah. Oh, yes. Okay. Yes. Yes. Thank you for bringing that point. So when the rest is not there on the membrane, it cannot simulate rest, simulate Huntington. So that property is required for it to have that sumo E3-like property. You're right. So I didn't bring that point. So rest has to have its interaction with the membrane to farnesylation to have this toxic property through simulation. No, what we have, this rest already has the COX domain, endogenously expressing COX domain, and the farnesylation happens on the cysteine of the COX domain to bind it physiologically to the membrane. So we didn't do any other way of bringing out to the membrane because it naturally goes to the membrane. Already it goes to the membrane. But when you have mutant, it cannot go to the membrane, cannot simulate, cannot promote toxicity. Okay. Now I just explained cell culture model. I'm going to go to the mouse model. The question is, does rest deletion has any impact on Huntington disease? So there are many different kind of Huntington disease models. What we took is an N71 model. This model, the Huntington disease, they die around 6 to 7 months. Prematurely, they die. Then what we did is we deleted rest from this mice, and then looked at what happens to the behavior and the pathology. So here I'm just giving you an example of like we looked at the motor effect on the rotating rod. Many of you know the mice can stay on the rotating rod and learn, and the wild type mice beautifully learn. But the Huntington disease mice, which is shown here, they cannot perform well on the rotating rod because the striatum is not properly functioning. But when you mutate, when you remove rest, for example, here 2 copy and 1 copy, they start performing much better. So no rest, it's better for Huntington disease mice. And then here is a lateral ventricle enlargement. This is what we see, enlargement of lateral ventricles in Huntington disease mice. But when you remove rest, 2 or 1 copy is sufficient. You can bring back the phenotype like wild type. So this clearly establishes that the rest has this toxic property to promote Huntington disease. kind of phenotype. Yes.
They have this sporadic movement also, besides the performance of the roles, they also move like something control.
The mice? Yeah. So what a lot of human behaviors we cannot reproduce in mice, right? So there are certain behavioral sets that you can mimic. They mimic striatal deficit. One of them is rotating rod or open field test and beam walk test that also can be done. But um, when they have a severe deficit, for example, they have a clasping, hind limb clasping. That's one of the major thing you can observe when the striatum is not functioning. So they really don't have a like chorea kind of. What you saw the patient with a chorea kind of choreatic movement. I don't think we see that in human. I'm sorry, in mice model. Yeah, that's a big thing. Another thing is like mouse models are not like human models. Yeah, the choreatic movement. We don't know whether there any mice can have a choreatic movement at all. I'm not sure about that. Yeah. Exactly. So yeah. So yeah. So there is, but interestingly, the mice model of HD they do not mimic choreatic movement naturally. Yeah. I see. That's a good point. Dyskinesia. Yeah. We did a lot of work on that. Okay. So let's move on to the next point that I wanted to bring. The question is if rest is sufficient to cause toxicity in the striatum. Then the next question was, what happens if you put rest in the cerebellum, which does not express rest normally, and then see what happens. So to address that, what we did is we took Huntington disease mice, we injected adenovirus expressing rest in the striatum, or just a control virus, and then we just want to see what happens. I'm going to show you some video here. Here is Huntington disease mice. They're around 3 months or so, I think around 3 months, and they do not have any major symptoms. And this is the surgery to inject control adenovirus in the cerebellum. So then we had a Huntington disease mice, similar age. Then we injected RES here. You can see these mice, they already have a very difficulty walking and very difficulty in standing, balancing. They're not able to stand there at all much. So then the onset of this phenotype appeared like within few weeks. So that we brought it back later. So that the clear and we used all kind of controls here. I don't have time to go to all the controls, but the idea is that there is if you bring RES and Huntington together, it's bad. When you look at the pathology in the cerebellum, like what is happening in the cerebellum, so we can clearly see the parvalbumin signal, which is one of the major neuron population in the cerebellum, is almost diminished. This is the quantification when you have RES and Huntington. And then, more importantly, when you looked at the aggregated form of Huntington in this mice, you can clearly, these are the control. This is the soluble form in the HD, no RES. This is a soluble form of Huntington in presence of RES. So there's a clear in vivo effect on the increasing the soluble form of Huntington when you have RES. So we think that because of that enhanced solubility may be killing the neurons much faster, more efficient. Okay, all in all, so far the model is that why striatum degenerates in Huntington disease. We have a very simple model. We think that our model supports that rest in the cerebellum, in the striatum interacts with Huntington and stimulates Huntington, and then Huntington now gets more soluble, and then which promotes cell death. So we have evidence that work from Uri, who did beautiful work, where we deleted SUMO1 in the brain, in the whole body, and those mice perform much better. Substantiating this pathway might be operating in the striatum. And that work we published very recently. That's the Uri's work here. Beautiful work with a lot of information supporting the model that the SUMO or REST, they act in the same pathway. And also what happens downstream. We have worked on that. Okay, what is happening after soluble form? And one of the pathway we identified could be mTOR pathway is potentially involved as a major regulator of Huntington effect. And also we identified that there's the ribosomes also may be involved in accelerating the disease pathology over time. Well, this was great. And the important point is this model is also supported by many different studies, independent studies. We are very happy. There is a substantial support, but something happened. That something happened was that we started seeing that this REST, this is a fluorescent REST, it starts forming this long tube. And then it starts moving into other cells by forming this membranous tunneling nanotube. It's called tunneling nanotubes. So what are tunneling nanotubes? So they are dynamic cell-cell interactions. They provide a new method of cell-cell communication. They play important role in intracellular exchange of signals, chemicals, organelles, pathogens, etc. As you can see, the field is growing. It's a very new field. A lot of research is coming up now, really substantiate its role. You can clearly see that happening here. But the question that we don't know is that what does the TNT really exist in the brain? You know, we have so many connections already, the chemical connection, electrical connection. So do the TNT-like structure exist in the brain is still unknown. That's where I think we have some data to support that. So little bit more on the G. So this is the cells which just express GFP. We don't see any much too, but when the cells have a GFP rest, you can see they start forming long tubes, and they resemble cytonyms in Drosophila, and they also resemble like TNTs in PC12 cells. And this is a scanning electron microscopy of TNT made by REST. You can see that it's a beautiful transition and the seamless transition from cell to cell. These are two different cells connected. They're membranous and they're positive for actin. If you prevent actin polymerization, the tubes cannot form. So it depends on this actin cytoskeleton to make it. Here is the electron microscope. You can see when you express REST, there are many tubes formed between the cells, but we don't see that in the control alone. A very robust formation of this tube. And because it's very new to the field and we want to a REST, we want to test whether Drosophila REST can do the same thing. Drosophila REST has a lot of similarity, not 100%. But we tried expressing Drosophila REST, as you can see here. Since the Drosophila REST also can form a beautiful long tunneling like nanotubes interacting with the neighboring cell. So why do this REST makes this tube? And what is the purpose of moving from cell to cell? And is unknown, is so far unclear. I'm going to show more data on that. And more importantly, it looks like this pathway seems to be conserved in Drosophila REST, also in mammalian REST. So there seems to be something going on. We did more experiment. We developed different kind of cells, stable cells, inducible cells, and human iPSC cell line. What we see is consistently, wherever you have REST, you have the formation of tubes. You can see it. It likes to make a connection and then interact with the other cell, getting transmitted. And that property again depends on the rest membrane binding. It has to bind to the membrane to make this. It's very specific effect. And here, because there is endogenous rest in PC12 cells, those are the cells first they demonstrated TNT, PC12 cell 2004. We depleted rest, and we found that if you deplete rest in PC12 cells, and they do diminish the formation of TNT. Again, this work is majorly run by Huri, who is sitting in the audience. The second, if I have any question, you should ask him, not me. So this is a beautiful work and a lot of data to go. So this you might like. Here, I'm going to show you a video, live cell image, how these tubes are formed, not how, but tubes are there transmitting cargo. And here, you have two kinds of cells. What we did is we expressed rest in the striatal cell, neuronal cell, and we expressed membrane marker in another striatal cell. Then we co-cultured them. We co-cultured and then did the live cell imaging. You can see these two tubes which are like a lot of tension there on top of it. And then they start delivering here. 1, 2, 3, 4. And then after some time, they start entering into acceptor cell. So they make the tube, they interact with the other cell and start delivering the cargo. Rest goes in, physically gets into other cell. So because it's the property of rest, we did a lot of domain work, which I briefly mentioned, like you have to have an intact rest to make this tube. You should have a full intact protein. If you can mutate many things, like membrane mutation, GTP mutant, they all make very less. The ligase domain, E3 ligase domain can have some effect, you can see here. And for effective formation of tubes and transmission to another cell, you need to have an intact rest. You need to have all the properties of rest. Okay, it's because this is a highway now. It's a highway, water can be transmitted through it, right? Here I'm going to show you some examples. Like here is a, sorry about that. One minute, I'll come back to that. Okay. Yeah. So what you see here is the TNT, and what you're looking at is endosomes being transmitted. The red is endosome, green is rest, because the endosomes are moving through from this cell to that cell. You may immediately think about, okay, why endosome? Because already the endosomes are other cell, right? Why it is transmitting endosomes here? We don't know. A lot of things we don't know. So, and here is the transmitting histamine receptors. So this looks like a guitar, right? With many strings. So you can see transmitting a histamine receptor from one cell to another cell. And here it is transmitting caveolin. Caveolin can move from cell to cell. You can see that one. Then after delivery, it detaches. Okay, what was the question? Sorry. Which drug, please? Oh, nitropropionic acid. It's a good point. What we did with nitropropionic acid is we didn't do that experiment, but what we did is we treated the acceptor cell with 3NP, 3-nitropropionic acid. We damaged the mitochondria. Then we co-cultured them with the healthy cell expressing rest. Then rest can form a tube and then interact with the damaged mitochondria in the acceptor cell. So we don't know 3NP can directly affect TNP, but If you damage mitochondria, rest can move from one cell to another, interact with the damaged mitochondria in the acceptors. Okay, the main point is that because we have shown so much about rest and Huntington link, we wanted to know whether these tubes can transmit Huntington itself. A lot of literature in the day, a lot of literature available, even in a human patient when they did cell therapy, cell replacement therapy, they injected a healthy cell into the striatum of the patient, and after postmortem, when they looked at the healthy cell had Huntington. That means it came from the patient. One example. They did in mouse models, the Huntington can go from disease cells into healthy cells. So there are a couple of other models exist showing that Huntington transmission happens. Because we have shown that the TNT formation is a link with the rest and Huntington, we want to see whether the TNT can transmit Huntington. Yes, you can see this is a TNT, this region. There are a lot of red puncta, they're all endogenous Huntington. They can transmit Huntington through the tube. And again, it seems to be specific. We don't see so much for the wild type Huntington. In this wild type Huntington is 7-glutamate. We don't see that so many punctas there. So the transmission of Huntington seems specific to mere Huntington. Here you might appreciate that. Here is the video showing that the red is Huntington. There's the Huntington and rest together there. That has been transmitted. You can see that using the tube. I have another one which my favorite. Here is the rest and Huntington, and they're together. You can see they already delivered a cargo with Huntington and rest together. They're trying to deliver another one. Punches. Sometimes so there's a clear delivery of vesicular structure, vesicular-like structure to the acceptor cell, and then the cargoes have been transmitted. So Huntington alone is not transmitted efficiently in this system. We have seen that. But for the transmission, it requires rest. So rest and Huntington go together. And here is that shown here. The facts quantification, how much is going, what is going. So I can show you that this is the Huntington and rest together. Every puncta that you see is rest and Huntington together in those cells. So normal Huntington is not transmitted so much, and there is a several-fold increase of Huntington transmission by rest. Okay, again, this is important, right? In research, you need to have a multiple evidences, multiple way to really understand your biology. Right? Go ahead. No, it doesn't happen. Here we don't see wild type Huntington in the tube at all.
So, you will assume that the form you can the one that is soluble.
Good point. Yeah, we don't know that point. We haven't established that concretely here.
Because a lot of people have been, I never liked that story, but think that the data would suggest that. So there are a lot of people who think that the Huntington is a consequence of protein aggregation.
It does form aggregate, yeah.
What you're showing is that.
Yeah, there are two schools of thoughts there. Because aggregates are everywhere in the body. Not all those regions die. But then the aggregated regions may have some kind of phenotype because you cannot have a cell with tons of aggregates. It may be having some kind of impact. But the degeneration part, when they seems to should have a soluble Huntington. That's what the idea is. Because there are also several people think that soluble Huntington needs to be there to interact with the toxic component. That was the idea. Yeah, but those is a common thing in the field because we can see aggregates easily. But we don't know what kind of species soluble form is causing toxicity. You might have heard of oligomeric species. We don't even know what it is. we don't understand that oligomeric toxic species. So, okay. Go ahead. Yeah. Please take, take your time. Yeah.
Since you already studied the aggregated form of Huntington, you put the fluorescent protein in both aggregates between the fluorescent. There's no change. And, and
Oh, sorry, I didn't get the question. Can you reframe it? I'm
I was
sorry.
wondering if by looking at the fluorescent emission, you can think the aggregate.
I see. Yes, absolutely. There are ways you can go and figure out what is being transmitted. Is this really aggregates or soluble form? But we didn't put, I think that's the important point. We need to really do experiments to really go there. There are also antibodies which detect aggregated form. We can just try to do the immunostaining to see which one stains up in that. Yeah. Oops, that's corrupted. Okay, then is a part of the Uri story I'm talking about. I think if you don't, any of you don't know Uri is just sitting on the second bench, second row, catch him. So Uri's question was, okay, all this fine staining, does it really happen in the brain? This kind of transmission. That was a question, right? So then we had to design a new tool for that. In biology, you need to have a different technique tools to really address complex questions. Without tools, we cannot be doing any experiment. So here, what we did is we developed a construct where you can induce rest in presence of Cre recombinase. So the idea is that whenever you have a cell, in this case, you express a Cre on rest through virus, and then you induce them in the cells where there is a Cre. So we can point out which cell expresses rest, which cell does not express rest. Because you have to, if you're looking for a transmission, you need to know where you are expressing cells. Otherwise, you wouldn't know. So we made this tool, and then we first wanted to know whether rest can induce TNT in its natural environment, which is medium spiny neuron. So here is the D2 neuron. These are normally the neurons look like when you put a EGFP on them. Nice processes. But when you do that with the rest, you can see now these neurons, they look entirely slightly different, although they have intact cell body, but we can see a lot of long tubes coming out. That kind of enlarged here. So even the neurons making these long TNT-like structures. And they can come from the cell body, they can come from the dendrites. So it has this function retained in most of the cells. Then first thing we did is, we made a mice where we had two kind of neurons, D1 neuron, D2 neuron. Sorry about this image, it's not very clear here. So what we had this mice has got D1 or MSN, which is a red TD tomato, and they had a D2 or MSN, which is Cre. So we can express rest in D2 or MSN and see what happens. So here what you're looking at is a brain slices. Then we express GFP alone. We saw a lot of these processes. Not many of them are interacting with D1 neuron, kind of quantified here. But what we found is the rest, you can see, this again, we're looking at three-dimensional structure. Remember, when I showed cell culture, there are two dimensions. You can clearly see the long tubes. But a three-dimensional structure, one can expect the TNTs cannot be straight. It has to have some kind of movement. So what we found is these thin structures coming out of D2 neuron, where rest is expressed, interacting with D1 MSN. It's kind of quantified here. You can see that one, the same structure, a little bit modified to see. So looks like even in a 3D environment, rest can make the tube-like structure interact with the neighboring neuron. And this is a video taken by Uri. You can clearly see this is a D1 neuron. the D2 neuron, and this is D1 neuron, red color. You can see there is a co-localization there going on. So we think that this kind of possibility of rest making the tube interacting with a neighboring neuron, getting delivered, can happen in a three-dimensional structure, in a 3D slice. And then we wanted to really quantify that how much rest is getting into the cell. Again, remember, TNT you cannot see in vivo in the intact brain because we do not have any tool to go and image in such a deep structure. So what we did is we did this experiment in another kind of my D1R, CRE, and D2R EGFP. And then we basically looked at the expression of rest in other set of neuron like a donor and acceptor cell. Basically, what we found is that when you express rest in the striatal region, you can clearly see there is a rest expressed in a D1 neuron, or in this case, and then when you go from that region, injected region, a little bit further away, you can start seeing rest fitting into the neighboring cell, which is quantified here. Not only that, most importantly, what we found is when you inject rest in the striatum, that's where the CRE is expressed. But we also found rest in the cortex. We can see this is what is the expression. When Uri showed me this first time, I said, "Are you sure it's happening in the brain?" Yes, it's happening. So you put this protein in the striatum, you start seeing them in the cortex. It should not be there to begin with. We saw them in the cortex. So after establishing that the rest can move from one region between the neurons and from the region to region, we wanted to know whether it can also transmit Huntington, mutant Huntington. Again, for that, Uri designed additional tools. So here, what we did is important. So you make one mRNA which has got EGFP and mCherry, and then they have this P2S side. They can get cleaved within the same cell. Then, if in a cell which you are expressing one mRNA, you will have two colors, green and then red for wild type Huntington or mutant Huntington. Then you can follow where it is going, right? And then we took this construct, made lentiviruses, we injected them in wild type brain and in the striatum and the rest knockout striatum. This is the expression showing the similar expression. Look what we saw. So this is an injected site, mutant Huntington and EGFP, because one mRNA. They're expressed in same cell. That's why all the red is positive for green, you can see here. But when you go away from the injected site, 500 micron, you start seeing only red, predominantly red. That means what? This red came from that place, right? We didn't inject red here, it came from that place. That means Huntington is moving. And then this is a quantification. You can see the wild type mice, knockout mice. There's a clear deficiency. We don't see wild type Huntington itself moving so much. So we don't see much there. And this is the injected spot. This is a 500 micron away. Also, you can see. The problem is this kind of phenotype is diminished in the knockout, clearly. That's quantified here. So all in all, what this data suggests is that the Huntington can move from one place to another between the cell type, and that movement requires rest. Again, we looked at the cortex. So you can clearly see this is a cortical expression of Huntington. Should not be there because it should be there only in the striatum, because expressed in the striatum. But we see them in the cortical neuron, cortical cells. We don't know the neuron, most likely. And that is diminished in the knockout. So basically, again, this data support the idea that the Huntington transmission happens from the striatum to the cortex in the wild type mice, which is diminished in the knockout. We don't see transmission of wild type so much between the neurons. Because we saw there's a transmission of rest in the cortical neurons, we want to do another experiment. We want to culture cortical neurons in vitro and see whether the rest can move from cell to cell, the cortical cell. Again, we took kinase Cre and the EGFP neurons, we co-cultured them, then we put Cre on rest. We were surprised to see that most of the rest is present in EGFP neuron. It's kind of weird. It's kind of like a virus. It's kind of virus getting into a acceptor cell. That's what is shown here. There's so much of rest gets into a neighboring cell and very potent transmission. I'm almost there. Last slide. So again, there's something missing here, but I can make it work. I told you I began with the idea that the striatum degenerates. There's a striatocortical degeneration has been observed. We still don't know exactly how this whole process happened. What our data seems to suggest that in the striatum, there is a molecular change, biochemical molecular change is happening because of rest. And the rest can now make the tube between MSN and then start moving from cell to cell. And from the MSN, it can get it can go to the cortex through a ITNT, I call miniature TNT. So that's what kind of predicted here. This is a transmission of Huntington from one cell to another. The red one is Huntington. That may be happening there. And then when that happens, when these kind of things happens in the brain, that is why the striatum degenerates first, then the cortex degenerates first. And that could expand the specificity of the problem. Can you fund me? Can you fund me on that? Yes, a great question. We are currently working on that. I wrote a grant on it. It was not funded, but I'm going to go more aggressively with more data. We're currently doing some kind of screening work. We want to screen. We have a great facility for screening, and we have a high throughput assay we're trying to establish to really go for screening. It's a good point. Yeah. So I want to add one last point. I think this is important in terms of for the future scientists. We think that TNT is going to grow exponentially in future, hopefully. It could be an interesting structure that we kind of lost all these days because of this one reason I'll tell you. So we know that this beautiful paint sketch by Cahal happened because of Golgi. He discovered the dye, right? And based on this beautiful sketch, Cahal believed that the neurons communicate without touching. They don't, because what he saw is when he looked here, he never saw any of them connected. Oh, there's no way they're connected. There is no way they touch each other, right? But Golgi said, no, not neurons are connected physically. So he believed in that idea. So these two guys fought so much in early century, and both of them got Nobel Prize. But then over the time, because we discovered chemical transmission, we discovered electrical transmission, that the neurons called neuron doctrine became more prevalent. Now everybody works on that idea when they talk about neuron communication, presynaptic, postsynaptic connections. Very important, they're happening. But we lost looking at TNT because when you fix the brain with all the PFA and you destroy TNT, you cannot see them. We have shown that the TNT is very fragile. So that's why we think that in addition to the chemical and electrical synapses, the membranous connection are critical. And it could be fundamental because there's a paper came out during pandemic in chemistry, some chemistry journal. What they did is they made a artificial cell using lipids. They took the lipid, they made a primordial cell type, and when they made it, those cells they naturally make TNT. So they have a tendency to make TNT. There's a beautiful paper in 2019. Yeah.
How general? I know that now is some use that group that it's a competitive in X
Yes.
and general. I mean, how prion affect how alpha nuclein goes from those.
Exactly.
How beta amyloid. Okay. How general.
Yeah. So I think exosome is important happening. Exosomes are also transmitted. We think that the exosome may have, when the exosomes come out of the cell, where would they go? They can go anywhere. So we think that the TNT kind of communication could be more specific. It can interact with a particular like cell body of the neuron, or it can interact with the dendrites of the neuron, or other cell types. So TNTs may have a specific path to find, but the exosomes just come out and then go somewhere. It could be another way of modifying the brain function, which is important as well. But how they are, which is more dominant, which is prevalent, what context, I don't think we know. I don't think we know those details clearly because nobody studies, not many people study TNT and exosomes together. And they may have a different cargo, different way, mechanisms. So this, as I said, the field is evolving, and those are the questions we need to address in the future. We need to have that clarity. Okay, how prevalent that is when this happens. So this again comes with more tools. For example, we don't know TNT markers. There's no TNT biomarkers yet. That is limiting the field. And also, we don't have a strong imaging system to go inside and image these protrusions. So I think we will go there. We could now, we couldn't have image all this structure 50 years ago, right? We have this technology to see them now. In future, we'll have advanced technology to see. Yeah. The bottom line is we exactly don't know how these competing. Do they compete, or they're two different pathways? Okay. So I'm done. These are the people that helped me to come here. I'm standing here because of all these people. Without them, I wouldn't be doing anything. One thing I missed here is, here is Tapia. I was permanent chat. I forgot to put his name now. I can't forget him. He was a very important collaborator. And all these people, I can see Uri. Uri, I should say, is a great postdoc and had great people to work with me all the time. You know, in science, you can't progress if there is no good people to work with. You know, you perish if there is no good people. You progress if there are good people working with you. I'm glad to have Uri with me for a long time. And all the people collaborated. The money is important. And thank you, Elta Call, inviting me. Appreciate your time. And any questions? Yeah.
If mutant soluble Huntington is the one that it's transported between the cells, could you analyze this looking at the simulated form?
Yes. The simulation deficient Huntington moves very less. It comparatively moves very, very little. So that's why we think that there could be some role for sumo in transmission. Whether it is doing it because of the solubility or it is because of the other protein-protein interaction, we don't know.
Yeah, I will guess that because that is what has been observed with the simulation.
Exactly. Yeah. It's possible. Yeah. I'm pretty sure there's a significant component, a significant role for SUMO in transmission. It's possible that modification Huntington may be playing a role.
And you mentioned that the mutation was with SUMO1, but have you checked if it is simulated by SUMO2, 3?
Yeah,
because they make branches, and that is what I was wondering.
Yeah, I think it's a good point. SUMO2 knockouts are lethal. They can't use them for experiment, so it could be playing a role. But SUMO1 deficiency is sufficient because we showed that in our experiment, SUMO2 is a major modifier Huntington, but it also can be modified by SUMO2 using a different D3 ligase. So we couldn't do that SUMO2 study because of the issue. We saw a significant reduction of SUMO1. I'm pretty sure if you're deleting both may have a better compensation, but again, it's a lethal.
Thank you for the talk. Very interesting work. I have two questions.
Sure.
The first one is when Huntington is transferred from the striatum to the cortex, degeneration in the cortex would also depend on the rest, because you said the expression is much, much higher in the striatum than in the cortex, but it could be sufficient to induce neurodegeneration in the cortex, or how would it be?
Yeah, so that's very important. We think that when the rest is moving into the cortex and it's taking Huntington with it, the cortical degeneration is observed less compared to the striatum. So there's a clear loss of striatum, then the cortex degenerates, some part of the cortex. So it's possible that even a subtle change, but again, in Huntington disease, the cortex also has Huntington. So what is the point in taking Huntington from the striatum? That is the question, right? So this is a point we're not able to resolve. That means we need to identify the function of this protein when they're getting transmitted. And what do they do? It's possible that you cannot have I'm just giving a bad example now. You can take one apple, right? And you can take one apple after 5 minutes, but you can't take 10 apples, although it's good for you. Maybe if you continue to transmit a bad protein to already a certain level of protein is there, then if you accumulate them with addition of from somewhere else, if that might cause a homeostatic stress and then generate a different kind of malfunction. So, because we're not able to really quantify those kind of things, but those are the important questions. First, we'll have to identify the function. So we will be doing. We wanted to know what does rest do when they go there. I think we will eventually get answers. That's good. Yeah.
Thank you. And the other question is that there are other proteins with polyglutamine residues that can induce the neurodegeneration in other parts of the brain, as ataxin and others.
Yes.
So you think there are other proteins similar to REST that can help in the induction of neurodegeneration?
We always
polyglutamine proteins.
I think so. I think that question I've been asking, people asking me so many times. It's likely then why would in ataxia the cerebellum degenerate? We don't have answer yet. There must be something in those regions might be impacting the changes, I feel. But yeah.
But can REST interact with these other proteins with ataxin?
They can, it can transmit ataxin.
Okay.
But not as efficient as Huntington. We did quantification on that. But it can transmit ataxin too.
Okay.
Yeah.
Thank you.
Yeah, thank you.
Thank you. I have two questions too.
Sure.
The first one is at the beginning of your talk, you were saying that this complex is leading to death. So my question is, which cells are the ones doing these TNTs? Is the cell like prior to die the one doing this, or is there those cells that survive? Because you had some cells that even with in using this complex were still there. So
yeah, this is a very good point. See, the disease, most adult onset diseases, the symptom appears after 30 years of age, 30 or 40 years of age, they get the symptom, which is a motor symptom. It has been demonstrated that even before the first symptom, the striatum shrinkage is observed in those patients. So that means the pathology may have happened like long time ago, but the critical point that you needed to have the symptom is different, right? It quite be different. That is why the people think that in a juvenile Huntington patient, they have a longer expansion. They have an early onset, the premature death. Most of the children who have more than 75 or 100 CAG repeats, they die very early on. So it's definitely CAG dependent impact. Adult onset, they are around 46 or 45 CAG repeat. They get slowly develop the symptom. When does the TNT appear? We can only speculate at this point that transmission may be happening as soon as there is a recent Huntington, may be happening even during the development, but there could be compensatory mechanisms also operating side by side. But once the transmission reaches a critical stage, then the disease start appearing. But we don't know the time window. That means you need to do that in vivo imaging. So very complicated.
My second question is more technical. In those constructs you were using for expressing the reporter rest, are you using the endogenous promoter or you're using a promoter that's overexpressing the protein? And my question goes in the sense that probably if this phenomenon could also depend on the level of rest you're expressing in the cell.
Good point. Very good point. So in this case, we have done many different kinds. Like we have used this one. I think we used a normal high expression promoters, but we also have made a knock-in rest, expressing rest in a safe harbor locus, and you can clearly see rest makes TNT no matter what. But then we also had a construct where mutant cysteine 263 using under the same promoter, same expression level, we don't see TNT formation. So, and also like when you make a knockout, and then reconstitute you can get the TNT, but you reconstitute with cysteine 263 mutant, you don't get TNT. So there is a clear effect of rest level, whatever the level it is, it seems to be making TNT efficient. PC12 cells have endogenous stress. If you delete endogenous stress, TNT is diminished in PC12 cells. Yeah.
Very interesting work.
Thank you.
I'm curious about the cell death mechanism. Very long time ago, John Reed showed in animal models of neurodegeneration that none of the apoptotic inhibitors were able to reverse in any way or prevent in any way the cell death observed in these models.
Exactly.
But you show in your first slides that apoptosis is still believed to be an important mechanism for this. But then you show that the soluble form that is tumulated is important apparently for this phenomena that you are describing. How can you conciliate these apparent contradictions?
Yeah.
I mean, the selectivity of your mechanism, can that induce a form of cell death that is not apoptotic, or is, I mean, can you, have you studied that part?
Good point. Good point. In human postmortem tissue, people have observed apoptotic kind of cell death. Also, I think some people say kind of necrotic as well. Most mouse models do not make apoptotic kind of cells. There is no cell death at all. They have a shrinkage of the striatum and enormous inflammation, other kind of biochemical transcription changes, but there is no cell death. So we don't understand why. But there is a pig model. People are doing a pig model. They show some kind of degeneration. Yeah, there's a good point. Like what TNTs, if they are making and transmitting anything, what kind of or cell death they induce, if at all, do it in human, is a question that we don't know. We don't know. But again, whether we really need to kill the neuron to cause dysfunction, you don't need to probably. You can cause some cellular dysfunction, maybe prevent or alter autophagy, and then you probably cause shrinkage and then cause the symptom. But we know from the patient MRI that the striatum is gone. Physically, no, there's no physical presence of striatum. That means they are dying, then removed from the system. That is the problem in the field, because the mouse models, we do not have apoptotic cell death. Yeah.
If we don't have any more questions, thank you so much again for your wonderful talk.
Thank you.